Thanks everyone. My name is Jessie Huang. I'm a postdoc in Darryl Cotton's lab at BU. So today I want to share with you our research over the last few months, adapting our iPS cell-derived lung alveolar type 2 cell model to looking at SARS-CoV-2 infections. So Julio went over a very nice background of this, so I won't dwell too much on the slide, but as you've heard throughout the day, alveolar epithelial type 2 cells, or AT2s, are the facultative progenitors of the distal lung, and that they proliferate and differentiate after injury. We know that AT2s do express ACE2, which is a key host entry factor in SARS-CoV, as well as in recent literature in SARS-CoV-2 infections. So we know that they're very important in terms of looking at COVID-19. And so COVID-19 patients develop acute respiratory distress syndrome, or ARDS, which is thought to clinically come about after the virus gets far down to the distal epithelium, distal lung epithelium. And I'm showing you here on the right these panels from the Lancet from Seattle at the beginning of the pandemic, looking at autopsy samples of infected alveolar pneumocytes as well as macrophages on top, and also looking at virions in this EM images in type 2 pneumocytes. So with that, there is a need for human type 2 cell models of SARS-CoV-2 infection, importantly also for drug discovery and to look at mechanisms of action of how SARS-CoV-2 actually infects type 2 pneumocytes. So this is where our iPS cell-derived type 2 cell model comes in. As you've heard over the years and from the papers, we have iPS cell-derived alveolar epithelial type 2 cells that can be sorted to purity, as shown here by this over 90% surfactant protein CTD tomato reporter. They can be expanded indefinitely in 3D sphere cultures and also adapted to air-liquid interface cultures. So I'm showing here a slide from images from Killian Hurley's paper of these cells, which we have single cell data on, looking at surfactant protein C transcript, as well as his 8-gene set differentiation marker. The link is below in case you want to go and play with this set. It's also on our website. And just this year, Christy Abel from Andrew Wilson's lab has published on the generation of these cells and adapting them to an air-liquid interface culture. So what is done is that these spheroids are dissociated to single cells and then reseeded onto a transwell insert, and the apical media is removed such that that side is exposed to air. So it is now an air-liquid interface culture, and this is what the cells look like. And upon establishment of this ALI culture, we're interested in knowing how our cells, both in 3D and in ALI culture, express host viral entry factors such as these two that Julio has just explained. So here's some data looking at our 3D spheroid model. This is using data from Killian's data set as well, looking at day 15 primordial lung progenitors, early and late human fetal lung, as well as adult type 2 pneumocytes in comparison to the IAT2s in 3D. You can see that as compared to the adult type 2 pneumocytes here shown in pink, that our IAT2s in 3D show comparable expression of ACE2 and TMPRSS2, which are two key factors for host viral entry. So then what about our ALI culture? We focused primarily on ACE2 in terms of these experiments. We used an ACE2 antibody to show that in these IAT2s at air-liquid interface, there is about a 10 to 20% expression of ACE2, of cells expressing ACE2, and that ACE2 expression is localized apically, as shown by these confocal Z-stacks. We're also interested in looking at how a spike protein on the SARS-CoV-2 might mediate cell entry. So because when we started this work, we were obviously working BSL-2, so we were interested in using these spike pseudotyped lentiviruses to see how they might enter the cells. So as proof of concept, here is an experiment done by Rhian and Werder in Andrew Wilson's lab, showing that with both pseudotype lentiviruses for VSVG and the SARS-CoV-2 spike, that while VSVG lentiviruses seem to transduce the cells equally for 293T and 293T with ACE2 overexpression, only the ACE2 overexpressing cells are able to be transduced with the spike pseudotype lentivirus. In our cells, in terms of the IAT2s, we show that the cells are equally transduced with the VSVG pseudotype as well as the spike pseudotype, showing that they may be permissive to SARS-CoV-2. So then you ask, what about the virus itself? So we have these wonderful collaborators at BSL-4 in Elke Mühlberger's lab at the Needle in Boston University. So Adam Hume in Elke Mühlberger's lab does all of our infections. So I'm showing you here the protocol starting from day 0, which you know the differentiation protocol pretty well by now. After they are plated at air-liquid interface culture, the cells are handed off to Adam Hume to do the infections in which the cells are infected apically with SARS-CoV-2. And then our initial experiments are with staining of nucleocapsid protein to look for immunostaining as well as flow cytometry, and then also to look at the nucleocapsid transcript levels. So here comes the beautiful images, if my computer will work. We're showing you here that with an increase in multiplicity of infection from 0.5 to 5, you see a nice increase in SARS-CoV-2 N protein positive cells. This increase is also shown with time. So from 1 day post-infection to 4 days post-infection, here at the highest MOI, you also see an increase in N positive cells. And here's just a magnified image. We see something similar with our flow cytometry data. Here I'm showing you mock 1 day and 4 day post-infection with SARS-CoV-2 Here you see that whereas at 1 day post-infection, there is a 20% of N positive cells. By 4 days post-infection, there is greater than 60% N positive cells. This trend is also seen at the transcript level. So here I'm showing you here MOI of 0.0004 all the way up to 0.04 of one of our initial unpurified virus stocks, showing that there is an increase in N transcript levels with the increase in MOI. And then there's also an increase in N transcript levels based on time as well in culture. And interesting, at this 4 day time point, we also see an increase in fragmented nuclei, suggesting that there is cytopathogenicity, that there's cell death going on as well. So with this data, we're also interested in looking at global responses. With this data, we're also interested in looking at how virus sheds. So here's a titer of where the virus is released. It seems like more virus is shedding from the apical surface as opposed to the basolateral side. We also have some very great ultrastructural analyses of these infected cells. So here I'm showing you here a very nice virion with the distinct corona in the cells. And from the extracellular space, we also show that there are some virions here where the arrows are pointing that are around the tubular myelin. So this really does show successful infection of mature functional type IAT2s because these tubular myelin form when there is surfactant being produced. We also show intracellular presence of the virus, of the virions. So here I'm confirming that where there are lamellar bodies, there are also these viral inclusions in lamellar body containing cells, which is consistent with the infection of the human IAT2s. So with this, we wanted to look at look for more of the global response of the SARS-CoV-2 infections. So we decided to do bulk RNA sequencing. So when the cells are plated at air-liquid interface, they are infected again apically at the highest MOI. We've been using MOI of 5, and then we collected 3 samples, a mock treated sample and the 1 day post-infection sample, as well as a 4 day post-infection. And here's the PCA plot showing you here the variation in the distinct clustering of these triplicates in each sample. And just a volcano plot here showing you a 1 day post-infection sample versus mock to show that in this particular sample, there's an increase in the viral transcripts as shown here, which make up 15 to 30% of all the IAT2 transcripts. So now we're interested in how different genes are changing in these samples. So here are some Loess plots. The first row and a half shows some viral transcripts of interest. You can see spike here and also nucleocapsid protein transcript here, showing that all of these viral transcripts increase by the 1 day post-infection sample and stay up at the 4 day post-infection sample. Interestingly, you see type 2 cell program going down. So transcripts for surfactant protein C, D, A1, et cetera, are all decreasing over time. And changes in terms of ACE2 and TMPRSS2, that might be due to their roles in the viral spread that we haven't had time to go into for this particular project, but is of interest as well. And all of these data are available on the Shiny app link here. If you want to go in and type in your favorite gene to check, that is available here as well. So we were interested in what kind of pathways are upregulated. So here's a gene set enrichment analysis of the 1-day post-injury infection versus MOC, as well as the 4 versus 1-day post-infection. And what you see here is that in both comparisons, that the NF-kappa B signaling pathway is highly enriched, and that by the 4 days, there's also enrichment of the interferon gamma response pathway. And here again are both volcano plots, the 4-day, which I haven't shown before, but with some highlighted genes of interest in both the NF-kappa B pathway and the interferon pathways, showing you that most of those genes are upregulated in the samples. For our unsupervised hierarchical clustering heat maps, you can see that these are some selected genes in the NF-kappa B and the interferon pathways, and that in the NF-kappa B pathway, both the 1-day and 4-day post-infection samples show upregulation of those genes. In contrast, for the interferon pathway, it seems like those genes are only, those interferon genes are only up in the 4-day post-infection sample, suggesting that there is a delayed interferon response due to SARS-CoV-2 infection. With this data set, we validated mostly via RT-qPCR. So those data are coming up here. We show via qPCR that, again, the type 2 cell program is going down over time. And here are some of the NF-kappa B related genes, which are all going up over 1 to 4 days post-infection. And the last set are at the bottom are the interferon stimulated genes, or ISGs, which do trend up by 4 days post-infection, but are not significantly different. And we know that this response is both delayed and mild, because when we treat ourselves with other stimulants like poly-IC and interferon beta, those agents induce a much greater response of the ISGs in comparison to the virus at 4 days post-infection. So we want to compare this to some human samples. So here are some autopsy sections from both COVID-19 and non-COVID-19 ARDS patients with these slides showing prosurfactant protein C staining. So the typical staining for prosurfactant C is this nice cytoplasmic punctate of prosurfactant protein C. When we go to the COVID-19 patient, you see a distinct lack of pro-SBC stain. So there's a decrease in the number of type 2 cells. In addition, there are regions of type 2 cell hyperplasia where you see strings of type 2 cells lined up together like this, showing the abnormality. And here I'm also showing some H&E stains as well as cytokeratin AE1, AE3 stains to show the sloughing of the epithelium in the injured areas in the COVID-19 samples. And finally, for my last part of the talk, we decided to check try to use our platform as a drugs discovery platform. We know from the literature and research going on that there are multiple methods of attempting to block viral infection, and that can be blocking entry, replication, or to modulate immune response and enhance the antiviral response. And we used Vero E cells, E6 cells, which are African green monkey kidney cells, as our control. For these cells, if they're treated with the virus plus the vehicle control, you see this upregulation in the nucleocapsid transcript. And then the next panels with camostat are the TMPRSS2 inhibitors, which do not seem to do much in these cells. Conversely, E64D, which is the endosomal cathepsin inhibitor, seems to significantly reduce the N transcript, and remdesivir here is our antiviral, which seems to significantly reduce N transcript by 4-fold. So how does this look in our IAT2s? For remdesivir, again, you see this nice decrease in transcript. However, there is a difference when it comes to the other drugs. For camostat, the TMPRSS2 inhibitor, unlike the VRE6 cells, we do see a significant dose-dependent decrease in transcript. And conversely, for E64D, the cathepsin inhibitor, there is not much effect at all. So from this, it appears that in the IAT2 system, the SARS-CoV-2 infection is dependent on TMPRSS2, but not dependent on the cathepsins. So in conclusion, I've shown that we have taken iPS cells that can be derived into IAT2s and plated in an air-liquid interface culture that allows for apical infection of SARS-CoV-2. These cells are indeed permissive to SARS-CoV-2 as shown by immunostaining and also EM images. And upon infection, it induces a rapid NF-kappa B response, a delayed and dampened interferon response, and a loss of type 2 cell program. And we think that this platform is very applicable for drug testing experiments and projects, and we're happy to share these cells for other collaborations as well. And of course, I definitely need to thank a lot of people for this project. All of these 6 people at the top have been instrumental to this project. Adam Hume in Elke Muehlberger's lab is the one who has done all of the infections thus far in this project. Chrissy Abel has been instrumental in terms of generating this model, as you guys have heard in previous talks by her. And Rhiannon Werder has also done a lot of the experiments for this particular project. And of course, I thank the PIs, Elke, Andrew, and Daryl for their support and feedback and discussions. And of course, a lot of people who have contributed greatly to this project. And with that, I'm happy to take any questions.